Está gravando.
Ok. Nice. Buenas noches a todos. Bienvenidos al ciclo de conferencia de las Olimpiadas Nucleares de América Latina, Jacapao Latam 2026, un espacio que reúne a jóvenes, investigadores y profesionales comprometidos con el desarrollo de soluciones innovadoras para los grandes desafíos energéticos, médicos y tecnológicos de nuestra región. El Hackapower se encuentra dirigido por Patricia Weyland, Eliane Silva, nuestras directoras. También tenemos a Yadna Méndez como coordinadora general, a Juliana Méndez como coordinadora de Relaciones Internacionales y Medios de Comunicación, y quien les habla, César Granada, como coordinador del tema de Radioteranóstico. El día de hoy también nos acompaña nuestro parcero de la Oncidium Foundation, Efraín Perini, que nos está dando un soporte con el sistema. Es importante resaltar que el Hackapower Latam 2026 representa una gran oportunidad para fortalecer la integración latinoamericana a través de la ciencia, promoviendo el intercambio de conocimientos y el desarrollo de la propuesta que contribuyen al avance de las aplicaciones nucleares con impacto social, ambiental y humano. Y es en este contexto la conferencia de hoy que cobra una gran relevancia especial, ya que abordará los avances en Radiotheranostics y terapias alfa dirigidas, áreas estratégicas para el futuro de la medicina nuclear y la producción de radioisótopos. Estos conocimientos serán fundamentales para inspirar ideas y aportar herramientas técnicas que ayuden a ustedes, los participantes, a enfrentar y resolver los desafíos propuestos durante la competencia. El día de hoy tenemos el honor de contar con la participación del doctor Valery Radchenko, investigador científico en Triumph, profesor adjunto de química en la Universidad de British Columbia y embajador de la Oncidium Foundation. El doctor Radchenko es especialista en radioquímica y en la producción de radionucleidos terapéuticos para la terapia radionuclídica dirigida, TRT. Cuenta con una destacada trayectoria internacional en investigación y desarrollo de radioisótopos para aplicaciones médicas avanzadas. De esta manera, su experiencia y visión contribuirán con certeza enormemente a enriquecer este espacio de aprendizaje e innovación. Dr. Valery, it is a great pleasure and honor to have you with us at Hackapower Latam 2026. We truly appreciate your time and willingness to share your knowledge and experience with all of us. Without further ado, the floor is yours. Welcome, and please feel free to begin the lecture. Thanks so much.
Excellent. Yeah, thank you so much for the introduction. Sorry, my Portuguese or Spanish, it's not as good, but I will, even, my title, it's look long words and complicated. I will try to do my best to explain it's in the simple words. And first of all, quick question, can you hear me well and can you see the slides? All good?
Yes.
Excellent. Okay, cool. Let's go. And just for what I'm planning to do, I'm planning to present for about 30 minutes, and then there would be some time for discussion. So, and if you see any problems with the video or audio, just let me know. Okay, so I just want to start, and again, my talk is going to be about some of the promising radionuclides that we can use for medicine. So we can use it for imaging, but more important, we can use these radionuclides for therapy. And before I kind of dive into the how we can produce and how we use these radionuclides, I really would like to take a couple of moments and introduce the place that I'm working. So I work at Triumph. So Triumph is a Canadian national lab. And what's unique about Triumph, which is based in Vancouver. So Vancouver, it's a beautiful city on the west coast of Canada. And what's unique and why I really like to work at Triumph is because we have a setup as the big and advanced national lab, but at the same time, as you can see from the slide, we're governed by the different universities across Canada. So I think this is something that makes this place very unique because we can do something very risky and very scientifically relevant which can make kind of future directions of development of radiopharmaceuticals. So, and as you can see, we pretty much represented from coast to coast by a number of universities, and also specifically the Kapov University and University of British Columbia, Simon Fraser University, and Triumph was established in 1968 and deliver already more than 50 years of great science. The second point, which makes me really excited to be part of Triumph, is that even I'm going to talk today about this life science component, so production of medical radionuclides, but Triumph is not only about life science and medical isotopes. As you can see, it represents a really broad span of the different activities, including dark matter cosmology, nuclear astrophysics, molecular material science, nuclear physics, particle physics. So all this make a great kind of mix of different research that and also kind of interdisciplinary collaboration, opportunity for interdisciplinary collaboration that really can make really nice projects. Like, for example, we have colleagues who want to develop the detector for some of the particles that we're working with. The colleagues with accelerator design want to make a new cyclotrons that can be more suitable for production of medical isotopes. This is just a few examples of this very nice interdisciplinary activities that we have at Triumph. And, in addition to all this kind of nice interdisciplinary different groups, we also have a lot of different accelerators that we can use for production of radionuclides, including the medical radionuclides, but mostly all of them dedicated to fundamental science. And what we are working in life science division, so our division, nuclear medicine division, we are looking at the using all these facilities for producing some medical isotope, but at the same time, we don't want to take over Triumph. We want to just coexist and complement the all these facilities and just to find the coexisting use or coexistence with some fundamental science. And then maybe I can just make another statement for my personal reason why I'm working on the production of medical radionuclides and why I'm working in radiochemistry. So, I'm sure many of you and hopefully all of you know what this periodic table of elements is. And as you know, there's 118 elements in the periodic table of elements. And this is all the tools as a chemist that you have when you do your chemistry. And then this is the radionuclidic, this is the chart of radionuclides. So it's another version of kind of expanded version of periodic table, which represent the isotopes of the element. So, as you know, probably each element may have a couple of isotopes, and some of them would be stable and some of them would be radioactive. But this table have more than 4,500 of isotopes in this. So you can see there's a much more bigger version of this periodic table of elements. And I think what makes me excited to work in radiochemistry, because instead of only 118 tools you have for your research, now I do have more than 4,500 tools that I can use in my research program. So this is something that makes me really excited, and this is why I'm do the radiochemistry and do research what I'm doing at Triumph. Okay, so now let's dive into some exciting science about how we actually can produce radiopharmaceuticals for the radiotheranostic. First of all, we need to produce the radionuclides, which we can do with many different ways, but most commonly, we can use the protons. So it's accelerators, for example, that many of which we have at Triumph, or neutrons, which we can get in reactors. So this is the two main source of the medical radionuclides that we can use, and facilities that we can utilize to produce the medical isotopes. Importantly, also, when you produce your medical isotopes, the next step, you cannot just directly use it for the application in radiopharmaceutical science. You need to do radiochemical separation. So you take your target, bring it to the lab. Hopefully you have some hot cell and automated module where you can run your target, and then you can get isotopes, what you want, that can be used for medicine. And later again, you cannot just use isotope itself, because what you need to do, the isotope itself would not go into the cancer cell, for example, if you want to image or treat it. So we would need to create a radiopharmaceutical. So we need to combine this isotope with a delivery system that can bring this selectively to the cancer cell that we can perform the imaging or therapy. And then if we do everything right, this is going to be a result of our effort. So this is the example of the patient which was treated with actinium-225, and you can see the scan imaging. So this is the image performed with imaging radionuclides for the patient which performed the therapy cycles with actinium-225. So you can see on the image on the left, there's a lot of metastatic lesions in the patient, where you see on the image on the right, it's almost lesion and cancer-free. So this is really amazing results that we can get with using the different radiopharmaceuticals, specifically the one that emit alpha emitters and actinium-225. So this is to kind of summarize you the structure of the modern radiotheranostics. So you have some selective system that's going to bring it to specific targets. So we'll often talk about the cancer. So that would be antibody or peptide that are selective for the cancer cell. And then we would combine this molecule with the radionuclide, which can be, depending from the emission, emit the positrons that we can use it for positron emission tomography or diagnostic, or emit the photon that we can use it for single photon emission computed tomography. So this would be the diagnostic option. And then if this radionuclide emit beta particles, alpha or Auger electrons, then we can use this radionuclide for the therapy. So we're going to use the same compound, the same molecule, and hopefully the same chelator as well. So chelator is practically the organic molecule that should hold the isotope, and at the same time can be connected to the biomolecules. And this would give us the opportunity to do theranostics. So we can use same element or radionuclide of the same element to do the imaging, and then use the same element but different radionuclides to do the therapy. It can be done with different radionuclides as well, but then we're going to use the radionuclide for imaging, and then we use another radionuclide for therapy. So this is the concept which we try to develop appropriate portfolio of radionuclides for. And then let's dive into the different option for the therapy, because this is something really now very important in the field of nuclear medicine, that we not only can diagnose the different disease, but we can really treat the different disease with the radionuclides. And then, as I mentioned on the previous slide, you have different options. You have alpha particle, beta minus particle, and Auger electrons. So if you see on this image, so if there is a radiopharmaceuticals, so the same what I showed you in the previous slide, just to kind of depict it in different format. So it would be selectively go to the cancer cell, and then depends from the emission, it would have a different length of penetration of these different particles and different damage to the different number of the cells. So if we talk about the beta minus emitters, the radionuclide that emit beta minus particles, then in this case, you would have really long pathway. So because of the low linear energy transfer, it will go millimeter distance before it would release all its energy. So this is not really suitable for small metastatic cancer, but would be suitable for the large cancers. So in contrast, alpha particles and Auger electrons, they have high linear energy transfer. So that means they would travel relatively short distance. So for alpha particles, we're talking about only dozens of cells, so micrometer distance. And for Auger electrons, we're talking about nanometer distance, which even not gonna cross the cell diameter. So I think this gives us the unique opportunity that we can use this emission as a very selective treatment option for the different disease, for example, cancer. So what's the problem now? Why we don't use them clinically very, in all the hospitals? So I think the main challenge is the availability of these alpha-emitting radionuclides, because often this radionuclide is located in the higher upper part of the chart of radionuclides, so having very high Z number, and they would be not easily available and produced. So as you can see, there's a list of different alpha emitters. So for example, radium-223 is only now approved alpha-emitting radionuclide, which used for treatment of the castrate-resistant bone metastatic cancer, because the radium chloride just naturally it's a bone seeker. Actinium-225 and bismuth-213, this is the same decay chain, and actinium-225 is one of the most developed radionuclides for the targeted alpha therapy. But there is also astatine-211, which represent a great interest to the community, and it's also became more and more available. Same as the bismuth-212 and lead-212, this is the system can also be used for the targeted alpha therapy. So all these three radionuclides are currently in their clinical trials. So there's no yet approved drug based on any of this radionuclide, but they are actively explored in the different phases of clinical trials. There's also some interesting alpha emitters that are only in preclinical trials, for example, uranium-230 and terbium-149, but they are still only in preclinical application. So if you're interested to read some more details about any of the slides, you can find often there's information at the bottom of the slide with the publication that kind of talk more about all these details. So feel free to look at these publications and get more information on all the kind of points I mentioned through my presentation. Okay, so let's now, because in interest of time, let's focus more on actinium-225. First of all, because it's something I have experience with, so I can more confidently talk about this. And also at Triumph, we're really advanced with the production of this alpha emitter. So first of all, as I mentioned, as for many other alpha emitters, it's really difficult to produce it. So what I mean by that, for example, there's realistically only 4 different ways that you can produce actinium-225. So one of them is decay of thorium-229. So thorium-229 have 8,000-year half-life, so really long half-life, which constantly decay to radium and actinium-225. So you can have so-called generator system. So you can use this long parent, keep it somewhere in the lab, and then elute every couple of months your actinium-225. So it's all good, except that there is only limited stock of this thorium-229 available in the world, because as you can imagine, it's not so simple isotope to produce. So with such a long half-life, so the current demand on actinium, it's much more greater than the availability of this thorium-229. So it's simply not enough. And that's why different groups and research labs start exploring the different way how to produce more of actinium-225. So another route that we can consider is the spallation reaction. So you practically have a thorium-232, which is still radioactive, but very, very long-lived. So it's 1.3 to 10 power of 10 years. So it's really, many, many years of the half-life, which you can make a target from and irradiate them at very powerful facilities with the protons, for example, at Triumph. And then you would produce actinium-225, but you also can produce the radium, which would decay to actinium-225. So this is something that we want to use, because together with actinium-225, you also produce another actinium-227 with a 22 years half-life, which can really be a challenge for the clinical application. So what we're really focusing on is getting actinium-225 from decay of radium that is produced by the thorium spallation. And then I mentioned there's 2 more methods. One of them would be irradiating the radium-226, which again, it's very radioactive target. As you know, 1 gram of radium is 1 curie of activity. So with cyclotrons, so in this case, you don't need such a powerful machine, like I mentioned, for thorium spallation. So you can use regular medical cyclotrons, but then the problem is to make a good design of your target, because it's radioactive, and it's you need a relatively large mass of it. So you need to be very careful and very kind of wise on the design of your target and the process. So, but then you can usually use the medical cyclotrons, and by P2N reaction, convert your radium-226 into actinium-225. Or you also can use the photons, and then you can do the photonuclear reaction. So you would introduce the photon, and you would kick out the neutron. So you would convert your radium-226 into radium-225, which also can be used as a source of actinium-225. And I'm very proud to say that at Triumph, we have a capability to do all these methods in-house. So we really have a lot of experience of producing actinium-225 by all these routes through in-house capability or within the collaboration. So, and I just give you a couple of examples. So we started about 10 years ago now to collect some actinium-225 with our mass separation facility. So again, I don't have time to explain all details of how the mass separation work, but again, you're welcome to look at this paper that, or two papers we published over the last couple of years. An idea is that we can actually, with our facility at Triumph, we can produce preclinical small quantities of very clean and very nice actinium-225 that we can use for establishing the measurement procedures, based protocols, so kind of for training purposes practically, and some preclinical application as well. But we not produce really a large amount with this method. So this is how we started about 10 years ago. Then we have a couple of collaborations that we get actinium from other sources, for example, from thorium-229 through the collaboration. And then since probably 2020, maybe 5-6 years ago, we start using main cyclotron, so our main 500 MeV cyclotron at Triumph to spallate the thorium reaction. So this is the second method I mentioned to you when I was going through the different production routes. So in this case, again, we produce actinium directly, so we can produce 225 actinium, but it's contain actinium-227, so which we don't want to have for the clinical application. So we use radium-225, which we produce as well, to generate actinium-225, which is, we call it isotopically pure. So this way we can really produce really nice large quantities of clinically relevant actinium-225. And our colleague at Triumph, we have a production team led by Ching Miao that doing this on a routine basis. Maybe I can just briefly mention this. I'm not going to stop and kind of explain in details, but and again, you're welcome to read all these publications. But also, I mentioned to you in the beginning that for to attach your radionuclide to different molecules, you need the chelator. So this is organic molecule. I mentioned that uses a bridge from the radionuclide between the radionuclide and the molecule. So in this case, at Triumph, we also have a lot of research ongoing for the different chelators. So this is kind of timeline. Again, we started about 10 years ago. And we, in collaboration with Cornell University, we developed this chelator called MacroPA, which is now very popular for using actinium-225. But then we also have a couple of collaboration in the University of British Columbia and also internally at Triumph that we develop different chelators that can really strongly bind actinium, and then we can attach it to different molecules for application in radiopharmaceuticals. So also, now let's look at the situation in the world about, I tell you that we really like at Triumph that we have many ways that we can produce actinium, but what is the situation globally? So as you can see, this is a very nice publication by my colleague Mariana Tosato, and you can also look at the situation for other radionuclides, and I think I have it in the next slide. But as you can see, there's a couple of centers and companies in North America and Europe, in Asia and Russia as well. So, but the current demand for actinium-225, it's the current availability, it's actually 63 gigabecquerel a year, and demand is already approximately 3 times higher than the availability. In the future, actually predict that we need even more. So I think we need to actively utilize all these different methods to produce more actinium-225 to be useful for the clinical application. And one important point, and I think it's relevant to this audience, what I want to show. So in this publication, as I mentioned by my colleague Mariana Tosata, who is currently working actually with us at Simon Fraser University and jointly with Triumph, we make this map, Alpha Atlas map, and you can it's open access, so you don't need any subscription to the journal to get it. So you can see availability of different alpha emitters. So it's the same isotopes I show you in the first slide with the availability and demand. And then you can see the color codes in different parts of the world. But what I really want to stress, that if you look at the southern hemisphere, other than a little bit of Latin Australia, there's not really much availability of alpha emitters, which I think is really important to note and change it. So I think what we're doing, we're actively looking into ways that we can introduce different alpha emitters into all these regions. So for example, 2 weeks ago, there was a fantastic conference, actually targeted alpha therapy symposium, which is the biggest event for the alpha emitters in the field, actually took place in Rio de Janeiro in Brazil. So this is really one of the steps that community taking to ensure the availability of these radionuclides across the globe. And also, of course, our colleagues at International Atomic Agency are doing a lot of effort to make these radionuclides available across the globe. So this is very nice publication, also led by another colleague, Jan Kleihans, who is just to start her professorship position in Prague as of last week. And this is very nice overview of different activities from International Atomic Agency about different events and trainings and schools and other technical documents that took place since 2013, so more than 13 years ago now. And what we're also looking, so I just want to kind of a little bit broaden your perspective. So this is the state of the art I show you on different alpha emitters, which is now key component for Radioteranóstics. But I also want to show you some of the new isotopes in new radionuclides that can be considered to be used for targeted alpha therapy. So we published last year, we published this fantastic publication where we screen all the different part of the chart of radionuclides, which present different alpha emitters, and then how you can produce them. And this actually, gives us another opportunity to see if there is other alpha emitters that can be used for the targeted alpha therapy in the future. And what I also want to mention, because I cannot not mention this, because this is the main topic of my research program, there's also other emissions that we discussed before that can be used for the radionuclide therapy, in particular Auger emitters. So, and as I mentioned to you, that these Auger emitters can be really very powerful because they only targeted single cell that we deliver it to. So in this case, we really have very nice selective option for the therapy on the only cell that we try to target, right? So this is from all three, beta minus and alpha would be the most selective option for the therapy. And what is also very nice, because we don't need very advanced, high energy cyclotrons or reactors to produce many of those Auger emitters. So you can use small to medium energy cyclotrons, which is available in many hospitals. There's definitely few in Brazil. We see when we visited 2 weeks ago, there's many in Argentina, and I'm sure there's all the countries in Latin America and Africa, and of course other parts of the world also have availability. So there's more than 1,500 different cyclotrons are available across the world. So there's many opportunities that this facility can be used for production of the Auger emitters. And this table just represent some candidates that we are currently exploring. And I want to spend a couple of slides because I just tell you a lot of information about different radionuclides, how we can produce them. And I also want to highlight some of the tools that our colleagues at the International Atomic Agency develop to help you to navigate through all these radionuclides and their production capabilities. So one of the interesting database I want to share with you, it's a cyclotron database. So because as I mentioned on the previous slide, that there is a possibility that you can produce Auger emitters with the medical cyclotrons. And this is actually a database which show you that there's a lot of cyclotrons. As you can see, there's a number of cyclotrons available in Latin America, some available in Africa, and definitely a lot available in North America and Europe. And you can use majority of them to produce therapeutic radionuclides, which I think is very important. And this is what we're working on in my research program. Another valuable tool that I suggest you can consider to use or check on. It's a medical isotope browser, and this is very nice way that you can estimate the production of different radionuclides. Again, maybe not all of you are experts in production of medical radionuclides, but it's actually a very nice online tool that can allow you to predict and estimate production of different radionuclides, which I think I use every day, and it's very helpful. So I definitely suggest you to check on this one. And another database, as I mentioned, you can also use reactors to produce the medical isotopes. So you can also check availability of different reactors around the world, and research reactors specifically. And you can see there's some summaries with more than 800 reactors is available, how many operational, how many under construction. So there's another interesting opportunity and database developed by our colleagues in the International Atomic Agency that you can use for some of these ideas, to get idea about the production of medical isotope availability in your region. And finally, I have another slide about the database. And I think this is very important slide that I want to share with almost all my presentations, because this is the user-maintained database. So if you are aware of any radiopharmacy around in your country, so you can see there's not many entries. There's only 34 countries and 64 institutions. Of course, there's many more now available in the world. So we want to encourage, and this is very relatively fresh database. So this is something that I encourage you to look at and see if there is a radiopharmacy around you. If not, one, please enter the information into the database. Finally, I also want to highlight a very nice event which is going to happen next year. So also organized by International Atomic Agency. It's International Symposium on Trends in Radiopharmaceutical, which is very important event in our field, and this happens every 4 years. So last time we have it 2023, so next one would be in 2027. So something to look at about the possibility to attend. And all the conference in International Atomic Agency is free of registration, and often the EAE also offers some stipends for member states to travel for countries that cannot afford it. So finally, just I see we slightly over time, so I just want to summarize instead of just a conclusion words. I just want to show you that our triumph code kitchen. So this is the periodic table of elements, but then you see surrounded by many different isotopes that we are producing. So the sum of the isotopes and radionuclides I present to you in my presentation was only a small portion of all activities we're doing in our lab. So there's a lot of exciting activities going on. And I also want to thanks before I move to my final slide to all my colleagues who actually helping me day to day in the doing all these amazing experiments. And especially I want to highlight all the trainees, all the students and postdocs that currently working on a program with us in the life science division. So you can see some of the alumni, some of the people who is still active and working in our group. So, and I also want to highlight that we at Triumph, we really have a lot of opportunities for different programs, including the core program. So this is just exchange program for the students. We have a graduate program, master student, PhD student. We also have opportunities for the postdoc. So feel free to check with these QR codes about these all opportunities. And I welcome, if you share this information with anybody who's interested. We're always looking for the good students. And with this, I would like to thank you a lot for the opportunity to present, and I would be really happy to discuss with you and answer any questions. Thank you.
Thank you, Dr. Valery. That was so amazing, and you still invite us for some opportunities. We will really share that with the group. I don't know, Cesar, would you like to see the questions in the chat?
Yes, I can read. Thank you so much, Dr. Valery. Yeah, we have some questions for you. Your presentation was amazing. Your knowledge, well, is amazing. Well, the first question is, in your opinion, what type of installation for isotope or radiopharmaceutical production, maybe alpha, most countries in Latin America will benefit more in this stage of development that the radiopharmaceutical chain is at the moment in the region? The answer will depend on the country or you. This question is in this chat, right?
Yep, I can see.
Yeah, that is the first question.
Yep. Yeah, it's a great question, and it's actually tied very well with my recent trip to Brazil, because I think we talk about how we can make some of this radionuclide exported to Brazil or imported to Brazil, and apparently it's a big problem to get the radionuclide to Brazil. So, and we was thinking about the possibility to kind of using the existing cyclotron facilities across the Brazil and maybe other countries in Latin America, that we can advance the production of some of, for example, Auger emitters, right? Of course, they are already used for production like fluorine 18 for the imaging, but if we want to advance diagnostic program, I think medium energy cyclotrons can be a valuable source for production of many therapeutic radionuclides and diagnostic radionuclides as well. So, but of course, I think available reactors would be another source, and I think they are already well utilized for production of medical isotopes. It's not so easy to build a new reactor, right? Because it's need to be many years of planning. But using existing infrastructure with the neutrons, I think this can be another way to get local access to different medical radionuclides.
Thank you. Actually, in Brazil, there is a nuclear reactor that is being built or being under project development. It's a little late, but I believe it is going to come, which is what's the name, Cesar? Do you remember? IPEN's, I forgot the name, but o reator do IPEN, o multipurpose. Yes,
Exactly.
but it's a little late, but I believe it's a very good opportunity for Brazil and also for Latin America, maybe South America. We should maybe sell to South America or something like that. I hope so.
Yeah, but I think that it actually was really impressive. Like before, when we have the symposium, we also have tours of the different hospitals in Rio de Janeiro, and we visit two private clinics in first day on Monday, and then at the end of symposium we visit the governmental or the clinic. It's a National Research Cancer Center, and it was fascinating how many things going on. And actually, in fact, I think in this National Cancer Center we visit on Friday, they already use the actinium-225 treatment of the patient, actually with the help of the Oncidium Foundation, which was really impressive to see some of this contribution from Oncidium. And also, like, one of the first, I think there was another study in Brazil before with actinium, but this is particularly the second use of actinium-225 on Brazil.
Sure.
So if you want, actually, I can see the questions.
We can end this the next question. Yes, we have many questions.
Yeah,
Interesting. The next question is, Dr. Valery, what do you believe is the roadblock for the development of the radionuclide production for medical uses in Latin America?
Yeah, I think it's according to the colleagues in Brazil. Again, I don't have experience with many other countries in Latin America, but according to them, it's a regulation, and they say it's very difficult to actually kind of promote the use of the new theranostic drugs because there is practically nothing written how to use them. And there's often some perception from the government to establish some new protocols which can make things slow. So I think, maybe working closely with the local authorities, because I think it's also, I can see this as an opportunity, right? Because from one hand, think about this, if there's no regulation yet, in many countries you can do whatever you want. But of course, it's depending from your local authorities who can say no, like we don't want to do anything. So I think I can see this kind of, as the one of the roadblock, at least from my understanding from the talking with the colleagues in the clinics in Brazil. And again, I'm just talking about particular Brazil because I don't know other, there may be, a very different problem in other countries. Maybe you guys know better situation in other countries. But I think this is kind of one regulation. It's probably one of the biggest roadblock what I can see, which actually not only in Latin America. I think it's, there's many countries in Europe and North America suffering from that. And I think it's changing, but very slowly.
Nice. Nice. Well, we have more questions. And these questions is from Mexico, and let me see, it's from Mexico. I'm working with TAT and Actinium 2025. I have a couple questions. We have two questions from Mexico. Size Macropa is patented. If we conjugate new molecules to it, we are essentially changing its structure. Does this create a new chemical structure? Will it still be considered infringement on the original patent rights? This question is in the chat to Dr. Valery.
Yeah, it's a great question, and I'm not the lawyer to probably answer this question, but I think you can actually use, you probably cannot create, if you want to have a clinical approved drug that you want to sell, I don't think you can do it with a patent later, but I think in your local pharmacy and radiopharmacy, you can use Macropa for treating the patient, even if it's patented. So kind of maybe short answer, from my understanding, you cannot sell something based on Macropa as a new product because it's patented, but I think you can actually use this to treat the patient in your hospital. And if you make some formulation yourself, and it's good to go from the local authority, I think this can be a possible way to move forward. See, Daniel, you just connected with the camera, so maybe if you can elaborate, or if you need more details, I'm happy to try to answer this in more.
Thanks, doctor. My question is if I can modify the structure of Macropa, if it's okay, maybe for a better performance of the chemistry of the compound. I don't know if it is possible.
Yeah, and again, I think going back to because there's also a linker, right? So there's a lot of components that you can practically claim as the new, right? Because the kind of maybe the ring, the macro cycle ring would stay the same, but if you have a different linker that can connect the chelator to the molecule, and maybe if you have a new molecule, so I think there would be a possibility to claim it as a new drug. And again, it also depends from the patent, because if this patent is North America or worldwide, so I think there's also can be the opportunities that you even can use the same compound if it's patented, if the patent not cover all the world and only because mostly it's usually people do it for North America and Europe. So if you do it in Mexico, maybe there's a opportunity to do it even as it is.
Yeah, I don't understand. Thank you, doctor.
the second question was answered? No, right? Well, the second question is
okay. Yes, thank you. Thank you.
Since the recoil energy from an alpha emission is around 100 keV, which is thousands of times stronger than a typical chemical bond, I'm curious to hear your thoughts on the best chemical approach to handle this. Are we seeing better results by designing pre-organized chelators that might recapture the atoms? What, was the question.
Yeah, it's actually a very nice and very good point of the alpha emitters, because I think, Daniel, you're very right that one of the challenge is that many alpha emitters, they're not decaying immediately to the stable or stable element. Is going through the multiple decay chains and then stable isotopes before they became a stable element, bismuth or lead, right? So I think this is something that need to be taken into consideration when you design the radiopharmaceutical, because from one side you can benefit from that, because per one decay of actinium-225 you get 5 alpha particles, because there is a different daughters is going decay and emit alpha particles. But the danger is that if you don't have a proper system to hold it in the same place, all the other 4 alpha decay is going to be happening somewhere maybe you don't want it to be happening. And I think for this reason, it's astatine-211, it's so popular now, because it's only alpha emitter in all this list that decay immediately back to the stable bismuth. So this is some advantage. There's some advantage of astatine, even if the chemistry is complicated, but it's don't have this issue of potential redistribution of the daughters. At the same time, Daniel, I think what we're seeing in the clinic, we don't really see the any side effects when we use the peptides, and I think there's no really kind of overall agreement why it's happening. But I think the reason for that is what at least people are speculating is, as long as you deliver your actinium inside the cell, because this is what peptides are doing, and this actinium would stay inside the cell, and all the decay daughters, they would staying inside the cell. Because if it's in bloodstream, like if you use antibody, then there's some research that can see some accumulation in other organs, for example, kidneys, but I don't think we see the side effects for using the peptides. And I think kind of, the one of the theories is, as long as you deliver your actinium inside the cell would act as the kind of container for all the daughters, and all alpha decay would be happening in the same kind of vicinity to the cell. But of course, there's a lot of debate what exactly happening. But at least, what companies is now doing, they just say, we don't care what's happening, as long as the drug is working and we see the effect on the patient, like, let's move on. But I'm fully agree that this is something very important that we need to understand carefully, and before we start using alpha emitters for different applications.
Nice.
Thank you. Thank you for your answer, Doctor.
Nice. And the last question for this night is a question from Brazil. Dr. Radchenko, what are the current limitants for the deployment of actinium-225 in Europe and also in Latin America?
Yeah, thank you, Yadna, for the question. So I think it's availability. I think, as I mentioned, for most of the alpha emitters, and not only in Latin America but everywhere, it's very difficult. Like if you start now searching where to get actinium-225, you would have probably a lot of troubles to sourcing one, because I think it's not means that there is not enough production capabilities, but there's already so many commitments to the clinical trials. So all the big pharma companies like Novartis, Bayer, Roche, and others, they already have a really, long-standing contracts with many company and producers. So if you are just a researcher, it's incredibly difficult to find the actinium right now. So I think kind of probably my the first answer would be that the availability of actinium-225 is a challenge. But then I think there's some specifics to work with alpha emitters compared to beta minus emitters because of the symmetry, many decay not immediately to the stable daughter. So there's many kind of procedures that are slightly different or different from the common application of theranostic radionuclides like lutetium, terbium, and beta minus emitters. So I think there's a second limitation that there need to be sufficient training of the new centers that want to start using the actinium. And this is actually why we have this TAP meeting in Brazil, because I think it was, the goal was to expose the community in Brazil to the experts who hopefully can work together with the different centers across Latin America and help them to establish alpha labs. So, Jana, is it answer your question?
Yeah, that was the last question. Yeah, it's good. It's all right.
Well, I'd like to thank you so much again, Dr. Valery, and thank you to everyone that was here and for the nice questions. It was very dynamic. Thank you so much. You can still make questions in the group, and then we can ask Dr. Valery. Thank you so much.
Yeah, thank you so much. And feel free to reach me anytime if you have any questions or if you want to. I think I'm available. I think I have my email on the last slide, so I'll be happy to stay in contact.
That's great. Thank you.
Thank you so much.
Bye-bye.
Bye-bye.